All right, welcome to this explainer. Today, we're cracking the code on exactly how to secure those top marks in your biology A-level. If you're gearing up for the Cambridge Biology 9700 exams, you probably already know that just blindly memorizing the textbook simply isn't going to cut it. We have to be smarter than that. So today, we are going to look at the syllabus directly through the eyes of the people who actually grade your papers. Let's dive right in and totally transform how you tackle these exam questions. Okay, so picture this. It's a real mystery that plays out every single year. Why do students who know the textbook inside out still fall short of an A? I mean, they know their biology. They've flipped those flashcards until their eyes blur. And yet, the marks just slip away on the final paper. It's incredibly frustrating, right? Well, the truth is, the assessment framework heavily prioritizes analytical depth over just superficial breadth. The examiners are setting traps. Now, they aren't trying to trick you, but they are absolutely testing if you truly understand how to apply biological principles in completely unfamiliar contexts.
Take a close look at this answer snippet we've got pulled up here. The secret lies not just in knowing the facts, but in dodging those classic examiner traps. The answer reads, sugar concentration was 0.5, whereas the other was 1.2. Do you spot the mistake? Boom, it's missing units. Principal examiner reports consistently flag that failing to include units in your calculations, like molar concentrations in a non-reducing sugar test, leads to an immediate mark deduction. It is such an easy fix, but under the pressure of the clock, students just forget. You also really need to check your terminology against the specific question context, instead of just writing down a generic, rehearsed description.
So how do we fix this? The big takeaway here is shifting away from rote memorization and adopting a highly systematic approach. Enter the 4-step answer framework. This is literally your high-band exam strategy. Step 1, identify the underlying biological concept. Step 2, extract evidence or data directly from the stimulus material they give you. Step 3, apply scientific reasoning to connect that evidence right back to the concept. And finally, step 4, use specific examiner wording. I'm talking about those precise syllabus terms that are literally printed on the mark scheme. This simple framework turns a messy, rambling paragraph into a laser-focused, full-mark response.
Let's actually see this framework in action. Look at this comparison regarding penicillin. On the left, we have a flawed student answer that vaguely states, penicillin stops bacteria from growing. It doesn't work on viruses because they are different. Yeah, that is definitely not A-level standard. But now, check out the top band response on the right. It nails the exact concept, inhibiting enzymes responsible for crosslinks in the peptidoglycan wall. Then it provides the reasoning. This leads to osmotic lysis, and it explicitly answers the second part by stating viruses lack this cell wall entirely, meaning penicillin has no target. Concept, evidence, reasoning, wording. Honestly, it's flawless.
Now here's a massive game changer when it comes to exam strategy, the method mark lifeline. Remember, biology isn't just writing essays. You're dealing with math, data extraction, and statistical analysis. Examiners actually award marks for your logical process and clear working. That means even if you totally botch the math at the very end, like you punch the wrong number into your calculator and get a bad final answer, you can still get points. But, and this is a huge but, you only get these lifeline marks if your thought process is clearly documented on the page. Let's look at this breakdown of selective reabsorption in the proximal convoluted tubule. Showing your calculation steps here literally secures critical marks. Notice that missing working penalty on the right side? If you just blindly write down final answer equals 12.5 with 0 steps shown, and it happens to be even slightly wrong, you get a 0, nothing. But if you clearly write out step 1, step 2, and so on, you capture those method marks. This is also a great reminder to ditch the vague language. Instead of saying, stuff moves into the blood, you must specify that glucose and amino acids move via cotransport proteins. Precision is everything, guys. Moving right along, the most frequent warning we see across all past papers is what I like to call the generic answer trap. This is when you write down a beautiful, absolutely textbook perfect definition that has zero to do with the specific graph, table, or organism actually provided in the prompt. Examiners want to see that you can apply your knowledge in real time, not just recite it from memory. So let's contrast a vague, memorized textbook response against a really precise, context-specific one. Check out this analysis of meiosis. Watch closely how the generic answer completely misses the mark. The low-scoring attempt just says, meiosis makes gametes. Chromosomes have. I mean, sure, it's true, but it is completely disconnected from the stimulus data. Now contrast that with the high mark strategy on the right.
It explicitly pulls data right from Figure 2, stating chromosome number reducing from 2n equals 46 to n equals 23. It connects the concept of reduction division directly to the biological function of maintaining the species' chromosome number during fertilization. It uses the data to prove the biological rule, and that, for sure, is how you secure top band marks. So how do we consistently snag those elite higher order marks? Well, it demands a solid critical analysis toolkit. When you see command words like evaluate, the examiner is essentially daring you to weigh the evidence. This means you have to actively question the assumptions made in an experiment. You need to spot data anomalies that just don't fit the trend, identify limitations in the experimental design, and correctly apply statistical tools to figure out if the results are actually significant or just a total fluke due to chance. Let's put your brand new examiner mindset to the test right now. I want you to pause this explainer for just a second. Try to predict exactly which statistical test you would use if an exam question handed you categorical data, like counting frequencies of different phenotypes in a genetics cross. Think about it. Are we talking about a t-test, a chi-squared test, or a correlation test? Okay, if you guessed the chi-squared test, you are absolutely right. Hit the nail on the head. This slide perfectly maps out exactly when to use it. Looking at the top left of our statistical selection guide, you can clearly see that chi-squared is your go-to for categorical data and frequencies. You'd use a t-test when you need to compare the means of two sets of continuous data. And for relationships between two variables, that's when you use a correlation test. Mixing these up is such a common B-grade trap. If you memorize this specific roadmap, you will easily navigate any data question they throw your way. Here is another major red flag detailed in the Principal Examiner reports: cause and effect confusion. Biology is absolutely full of complex sequences, and students frequently lose points by incorrectly swapping the biological triggers with their outcomes, or just scrambling the timeline of events entirely. Plus, a lot of people accidentally substitute the biological why, like the evolutionary purpose, for the how, which is the actual physiological mechanism the question is asking for. To show you what examiners are really looking for, let's use the link reaction as our case study here. You simply cannot just write a messy, vague answer like pyruvate breaks down to acetyl-CoA. That totally lacks detail and completely confuses the sequence. Look at how the top band answer breaks it down step by step. Pyruvate is decarboxylated, meaning it releases CO2. And then it's dehydrogenated, which reduces NAD+ to NADH. Only after that does the remaining acetyl group combine with coenzyme A. You have to clearly map out the precise sequential steps to prove you actually understand the mechanism. And speaking of precision, let's talk about the dreaded terminology trap which loves to show up around enzyme activity. Looking at this bell curve, a super common mistake is saying an enzyme is inhibited by an extreme pH. Actually, scratch that. It's not just a mistake, it's incorrect terminology. Inhibitors are very specific molecules. The correct syllabus term for what an extreme pH does to an enzyme's active site is denaturation. You have got to distinguish between terms like denaturation and inhibition, and always frame your answer within the exact pH context provided in the prompt. So I know what you're thinking. How on earth do you remember all this under the ticking clock of a real exam? To help make all these procedural steps and complex terms actually stick when the pressure is on, you should definitely use retention tactics. One of the absolute best ways is creating your own mnemonics, using the first letters of key sequential terms. When your brain inevitably freezes in the exam hall, a simple, silly acronym can literally unlock an entire 4-mark metabolic pathway in your memory. Now, let's focus on how to pull evidence effectively using this selective breeding example. Notice how the top band answer on the right doesn't just list off the theoretical principles of artificial selection. Instead, they explicitly extract data right out of the stimulus material to back up their comparative biological claim. Basically, if the question gives you a table showing crop yields over 20 generations, your answer absolutely must include those specific yield numbers to justify your conclusion. The data is sitting right there on the page for you to use. Do not ignore it. Finally, let's look at this incredibly detailed slide on bioinformatics, which perfectly demonstrates the ultimate A-star strategy: the power of cross-topic connectivity. See, bioinformatics isn't just some standalone topic about computers. It's about massive databases of nucleotide and amino acid sequences. The highest-scoring students know how to link this data-heavy topic to completely different syllabus areas, like protein synthesis, genetics, and even evolution. Showing the examiner that you can connect these databases to broader biological principles is a proven method for demonstrating deep conceptual understanding.
All right, let's bring it all together. The crucial takeaway from this explainer is your mastery checklist. Before you even write a single word, systematically deconstruct the prompt to figure out what's it really asking. Execute your math with clear, step-by-step notation so you secure those vital method marks. Relentlessly avoid those generic textbook answers by anchoring your wording to the specific context given in the question. And finally, always verify your units.
Well, the mystery of why smart students lose easy marks is officially solved. It essentially all boils down to method, precision, and context. But honestly, knowing the strategy is only half the battle. The real question I have for you is, how will you apply this insider, deconstruct, solve, evaluate method to your very next practice paper? I highly recommend mapping out your next past paper answer using the 4-step framework we covered today, and just watch your marks transform. Thanks so much for joining me on this explainer, and happy studying.